Hello, everyone, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled Scaling PCR Workflows from Benchtop to Automation. When the PCR concept first came to Carrie Mullis on that lonely California road so many years ago, I wonder if he immediately grasped the wide-ranging applications of this revolutionary laboratory technique. Identifying genetic mutations, detecting pathogens, genotyping, subcloning DNA targets, and potentially predicting drug responses. The list goes on and on. In essence, PCR changed basic life science research forever. However, as wonderful as PCR is, it can also be a time-consuming and labor-intensive process. In addition, the method is susceptible to variation by different practitioners. The good news is that there is a solution. It's called automation, which will comprise a great deal of today's discussion. One of our speakers will even note that all of the chemistries involved in late PCR are automatable. Let's meet him and the other members of our panel. Dr. Larry Wang is Professor of Biology in the Laboratory of Molecular Medicine and Global Health at Brandeis University. Larry is going to talk about late PCR and single-tube sample prep. Dr. Greg Shipley serves as Assistant Professor and Director of the Quantitative Genomics Laboratory at the University of Texas Health Science Center in Houston, Texas. Greg will discuss the specifics on how scientists can actually scale PCR workflows from the benchtop to automation. Dr. David Noor, Applications Manager at Agilent Technologies, will look at the planning and considerations that provide the rationale for automating PCR workflows. I'm John Sterling, Editor-in-Chief of GEN, and I'm going to serve as the moderator. Please feel free to enlarge the slide images or download the complete presentation. At any time during the webinar, you can send in a question for our panelists. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many as possible during the Q&A segment that takes place after all the presentations have been made. I think we're set to begin, and we'll start our webinar with Larry Wang. Larry?
Thank you, John. I'm delighted to be here. I was here today to talk about sample prep for single-cell, single-tube late PCR, and I will explain as I go along what late PCR is. The first slide shows you that this effort of ours began in 1997 when we were interested in pre-implantation genetic diagnosis as well as early cancer diagnosis. And they're both cases in which you could potentially have to start with as little as a single cell and get it right. The next slide shows that we knew the limitations of a conventional real-time symmetric PCR, and they are really two. One is in the upper panel that as you add less and less starting material to the reaction, you typically see a decrease in the plateau value as it takes more and more cycles to generate a signal. And in the lower panel, you can see that among the replicates at whatever CT you go out to, there's a lot of scatter at endpoint as the symmetric reaction exits exponential amplification, and that's, of course, the reason why people have to resort to real-time PCR. Next slide, please. So what came out of our initial thinking was a whole systems approach to revamping not just sample prep, but the chemistry of PCR itself, and that's late PCR, as I'll explain in a minute. On the left, you see that we also invented a reagent called PrimeSafe, which improves reaction specificity, not just at the start, like hot start, but throughout the reaction. And we worked on sample prep, and we came up with PureAmp and Quantilize, PureAmp for both DNA and RNA, and Quantilize for DNA. And finally, most recently, we've come up with something called lights-on, lights-off probes, which unfortunately I don't have time to talk about today, but it's quite exciting. These chemistries together allow us to re-examine virtually all of the tasks that one would like PCR to do, from RT-PCR through sequencing and multiplexing and quantitative analysis, and then actually to get a great deal of information out of multiplex, we use a new set of probes. And you can access some of this information on our website at latepcr.org. Okay, the next slide illustrates the real fundamental difference between conventional PCR, which is in the upper left, which is exponential amplification, where a double-stranded product is generated and accumulates, and when it reaches a detectable level, we call that real-time PCR, and when the reaction continues to accumulate double-stranded, eventually falls out of symmetric exponential amplification, and that's when you get scatter among the replicates. Late PCR stands for linear after the exponential, and so the reaction starts out exponentially and generates double-stranded DNA. And then just at the time that it becomes detectable, it switches to linear amplification, so it generates just one strand of DNA, and that continues with very little scatter among replicates, as you'll see. And so it's the accumulated single strand of DNA that is very, very useful to have. Next slide shows you that using this whole systems approach, we first tackled the problem of sample prep for genomic DNA in single cells, and that led us to quantilyse. And the next slide shows you the basic problem. If you put a single cell into a tube, which is a diploid cell, you would anticipate that replicate cells would have the same amount of DNA and so would give you the same CT value. But in fact, what we saw early on was that we got a lot of scatter in the CT, and that suggested to us that the problem was that the substrate, the genomic DNA itself, was not readily available in all cells. If you go to the next slide, you see the answer to why that would be the case, and that is that, of course, genomic DNA is wrapped around nucleosomes and packed into chromatin at various levels of compaction. And so if you don't strip the DNA thoroughly off the protein or digest the protein thoroughly, you end up with differential behavior of the DNA in different cells. So what we did was we came up with a reagent. The next slide shows the reagent called Quantilyse, in which we minimized the scatter among the replicates. So Quantilyse gave us, for single cells, a CT value of about 34, plus or minus less than one CT value, whereas other conventional ways for doing sample prep were much greater scatter. And the real secret to Quantilyse is very simple. It's basically SDS proteinase K, where we took out the other things that people had put in, like magnesium, which stabilizes chromatin, and DTT or other mercaptanes, which inhibit PCR. We, of course, put magnesium back in later for the PCR amplification. So this enabled us to do digestion and then kill the enzyme and then continue in a single tube to do the amplification. And the next slide shows you the results of such a study. At the time, we hadn't invented late PCR yet, so this is symmetric PCR at the single-cell level for the Tay-Sachs disease gene, allele 1278. And what you see in green are the homozygous normal in the upper left. That's using one molecular beacon for detection, or the homozygous mutant in the lower left for the other allele. And on the right-hand side, you see what the two beacons give you for a heterozygous cell. So it's very lovely, robust, symmetric PCR, but it has low scatter at the CT, but immediately thereafter it scatters as the reaction decreases. So we were pretty satisfied with this until we hit upon late PCR. The next slide shows you what late PCR can do, and it shows you two things. One, in the upper panel, this is a triplex reaction. You can see that the CTs are now extremely tight in the replicates and continue tight for quite a long time until they splay out at the end where the dotted line is. Those are genomic DNAs that are basically intact, prepared with Quantilyse in the very tube that they're then amplified in. Now, if you freeze and thaw the DNA instead of just proceeding in the same tube, what you find is that the scatter increases, and specifically it occurs early in the amplification right after the CT. And the reason for that is that anytime you freeze and thaw or handle the DNA roughly, you generate shearing, and that shearing is prone to mispriming early in the reaction. So we use the scatter after the CT as diagnostic about the state of the genome that we're dealing with. Now, if you go to the next slide, you can see that building on that, we developed a reagent called PrimeSafe, and the use of PrimeSafe is to suppress mispriming. It doesn't actually function as a hot start, rather it increases the specificity of the Taq polymerase, so the Taq prefers to not start on misprimed primers. And that is functional throughout the reaction. And if you go to the next slide, we now revisit the triplex reaction on the top that didn't have any PrimeSafe and compare it to the same triplex reaction on the bottom panels where we've added PrimeSafe. And what you see immediately is that we've eliminated the scatter among the replicates all the way out to the very end of the reaction, which is really quite remarkable. If you go to the next slide, you can see that this property of the combination of late PCR and sample prep and PrimeSafe allows us to do quantitative endpoint analysis. Rather than measuring things in real time, we can simply run the reaction at high stringency and then quantify the amount of signal that we have at the endpoint, which is indicative of how much starting material is. So that's quite a lovely feature of late PCR. Next slide. To return to our whole systems approach, I want to draw your attention to the PureAmp on the right. That's our approach to how to prepare samples, single-cell samples for RNA analysis. When you go to the next slide, the way we think about this is shown here. If you were to be on the coast of California and standing next to two redwood trees and imagine one is protein and one is RNA and they're touching, if you cut them both down and throw them into the Pacific, the chances of that DNA tree and the protein tree ever bumping into each other again is very, very low. Well, similarly, in a reaction containing protein and RNA molecules, the difference between their size and 1 microliter is shown on the left bracket, and you can see it's, again, 5 orders of magnitude. So all you really have to do is separate the protein from the nucleic acid, in this case RNA, rather than actually clean it up and purify it to completion, which is the conventional approach using columns. So using that approach, we then developed, as you see in the next slide, our PureAmp method, and this is the work of Christina Hartshorn in my lab. And basically we put a lysis material, which we call a lysodot, on the cap of a PCR tube, and this, of course, could be automated. It could be set up with hundreds or thousands of PCR tube caps set up this way. And then we pipette a single cell onto that lysodot, and the lysodot contains guanidine isothiocyanate and other reagents, and the cell lyses instantaneously upon touching it, and all of the proteins are denatured by the chaotropic agent. And we then dry that briefly to further increase the concentration of the guanidine. And we can either store that or we can add it to a DNA mix if we want. We can then spin it down to the bottom of the tube, add an RT buffer, and do an RT-PCR either in 1 step or in 2 steps. That depends on how you design your reaction. So again, the overall approach is doing everything in a single tube with minimum number of steps. So in order to validate this kind of an assay, turn to the next slide, what we turn to is using the exist gene expression in mouse embryos. And what you see here from the literature is that in an 8-cell embryo, if it's an XX female, each cell of the embryo expresses exist, and you can see that in the fluorescent dots for in situ staining, and the same is true in a 16-cell embryo. But if it's a 16-cell male embryo, XY, none of the cells is expressing exist. So we took advantage of that biology in order to validate our assay. The next slide shows you that if you take genomic DNA from a male and you do a dilution series and then run a test for both the exist gene and the SRY gene. Now, neither of these genes is expressed in the male embryo, and here we are looking at genomic DNA. So we're seeing that there's one copy of each, and it's a nice linear plot of both genes in the duplex. And even if we add the denaturing solution that would be left over from our PureAmp method, it has no impact on the linearity of the dilution series. So that gave us some confidence that PureAmp was working for both sensitivity and accuracy. We go to the next slide, shows you a biological experiment that really verified that. Here what we've done is take blastocyst stage embryos, male and female. The males are in shades of blue, the females are in shades of red. And we tested them in the upper panel for SRY and in the lower panel for exist. The SRY gene, of course, is absent in the female, so the female comes up negative and is present in all of the male embryos, and so they all come up as replicates, but there's no SRY RNA, so we don't see cDNA there, we just see the SRY genes. In the lower panel, the male embryos, again in blue, have one copy of the exist gene, and so the CT for their exist values is the same as it is for the SRY, but in the female embryos, we see a shift to the left in the CT, and that's due to the composite sum of the two exist genes per female cell plus the exist cDNA that was made in our reaction from the exist RNA. So we can use those data to deduce the copy number of the exist RNA or mRNA per embryo in this case, and we can take that down to the single cell level as well if we want to. So we've used the biology of the system to verify the technology. The next slide shows another approach to this problem. We examined heat shock gene expression, and the reason we did this was we were using a laser to burn a hole in the zona pellucida of 8-cell embryos, and the question arose, does the laser cause heat shock damage? So we used the laser to burn the hole. We then collect single blastomeres. And if you go to the next slide, you can see in panel A on the left 3 different experiments in which we collected as many of the single blastomeres as we could and measured HSP70 heat shock protein in embryos that had previously been heated. So all of these embryos should be expressing heat shock, and indeed each of the cells was expressing heat shock mRNA. The green bar shows the averaged value of all the blue bars in each experiment, and the red bar shows the HSP70 expressed per cell when we heated the whole embryo and tested the whole embryo. So the fact that the green bar and the red bars are essentially equal shows that by summing the values collected in the single cell, we end up with the same value obtained from a whole embryo, which was very gratifying when thinking about the PureAmp method. Now, in order to answer the question that we set out to here, we tested the question of whether the laser shot caused heat shock damage to the cells itself, and so we didn't heat shock the embryo, we lasered them, and we asked, did the cell adjacent to the laser shot express heat shock, and that's cell number 1 in the series, and you can clearly see that no heat shock was expressed in any of the cells following laser ablation. So that also verified the use of a laser. Okay, so to summarize in the next slide, what you can see is that the way we've approached it, both for Quantitative and for PureAmp, for sample preparation, is the idea is to do everything in a single tube and then by dilution go on to the PCR. That allows a separation of components rather than complete purification. It's experimentally convenient. It's less expensive. You don't use additional tubes. It's quantitatively more reliable because you don't move things along surfaces and separate tubes, which is a big issue when you're working at the single cell level. Of course, it has a lower risk of contaminating your lab and a lower risk of contaminating the sample, and it's very gentle. It doesn't result in shearing, which, as I showed you earlier, is one of the causes for scatter among your replicates. And finally, the whole set of approaches we've been pursuing is fast and it's automatable. So I'll leave it to others in this panel to describe how robots work, but certainly everything we've done here could be adapted to robotics. And finally, the last slide just summarizes our core strategies, which start out with late PCR for generating abundant single-stranded DNA in an efficient way. Dilute and go sequencing is a technology we've developed for using that single strand directly for sequencing without having to purify it. Quantitative endpoint analysis I showed you briefly is one of the nice features of late PCR. And PrimeSafe, which cleans up reactions and prevents mispriming throughout the reaction. It also enhances multiplexing, which I haven't had time to talk about today. The accumulation of single-stranded DNA in a late PCR reaction then enables us to separate probing from annealing of the primers, and so we can now use low-temperature probes, which are either sequence-specific or mismatch-tolerant. And most recently, we've invented lights-on, lights-off probes, which enable us to do high-resolution analysis over an extended stretch of DNA, where we can see single-point mutations, every single-point mutation in essentially 100 or several hundred nucleotides. So that chemistry, too, of course, is automatable. It's what you put into the sample mix rather than anything else. And I will stop there and send it back to you, John.
Larry, thank you for that excellent presentation, and in particular your discussion of late PCR and the advantages of single-tube sample prep. What's more, you've indicated that many of the steps involved are automatable, which is the major theme of today's webinar. If you're just now joining our webinar, welcome. As I previously mentioned, we'll be conducting a live Q&A segment following the panelists' presentations. Again, please type your question for any or all of our panelists into the Ask a Question box on the lower left of your console, and then hit Submit. Our second panelist, Dr. Greg Shipley, will now begin his presentation. Greg?
Thank you, John. What I'd like to do is talk to you today about using automation for PCR, and particularly quantitative PCR. So on the next slide then, what do we mean by automation? Automation refers to using liquid handling robots for component assembly instead of processing a workflow manually, which is what people normally do in the laboratory. It can be as simple as aspirating and dispensing a liquid from plate A into plate B or into multiple plates or other vessels. It can be as complex as automating every single step of a complex workflow utilizing multiple instruments, and I'll show you an example of that in just a minute. Many different kinds of instruments can be integrated with robotic software, and you can put the whole workflow together in a totally automated fashion. Next slide, please. So this is an example of a robotic workstation that we have in our laboratory for our screening program. This is centered around a Biomech FX dual-arm robot. The first component you see on the far left is a Cytomat 1, which is used for bringing new tips or plates onto the deck or storing used plates and tips after they've been used. Using the little elevators, you can see sort of just to the right of that, the little black bar that goes across. The gray instrument that is to the right of the Cytomat tower is a DTX-880. It's a multi-mode reader for reading multiple plate assays and biochemical assays. Then you can see the dual-arm robot in the center on the left. The first arm is a 96-tip head, and it can pick up a whole box of tips at one time and aspirate and dispense into either 96 or 384 well plates. On the right is the second arm, and you can see that's a span 8, which means it can pick up individual tips or all 8 at one time. The instrument to the far right is what we call the Cytomat 2. It is a tissue culture incubator. It has CO2 and runs at 37 degrees, holds up to 42 plates at a time, and these are for using for our cell-based assays. There also has an ELX 405 plate washer on deck, but it's not shown in this particular figure. So this is a figure of what it looked like when we initially assembled that. We've moved to a new facility, and if you look on the next slide, you can see what the robotic workstation looks like now that it's complete. As you can see, it's now sitting inside of a Biocell HEPA-filtered hood. You can see the HEPA filters on the very top, and you can see the sliding doors that we use to gain access or to close off to keep an aseptic environment on the inside. And so all of these components then are used for our siRNA or compound screening program where we're using live cells, or even when we're using biochemical assays that helps to keep dirt and other contaminants out. So all these instruments can be controlled by the Biomech software or by scheduling software, SAMI, that we use as well. On the next slide. So why do we use automation then? So automation brings a level of accuracy and precision to an experiment that cannot be achieved manually for large numbers of repetitions. So if you have a small task, a single or maybe two 96-well plates, it's probably faster and can be just as accurate to do that process manually. However, if you're using automation, means that, if you have a lot of plates, after a while, the human thought process starts to break down, especially if the pipetting pattern is very complex, and I'll show you examples of that in a little while. So using automation means that every plate will be processed exactly the same every time, regardless of how many repetitions there are, how complex the layout is on the plate, or how many different plates are involved, or how long it takes to do the whole task. If we look on the next slide. So what's involved in using automation then? So aside from acquiring the instrumentation, which you might think is the largest part of the operation, in fact, learning to use the software in a very sophisticated way is a critical step in using the robot. And that matters whether it's a relatively small robot or one of the larger ones that I've shown you so far here. You want to be able to use loops, nested loops, if statements, variables, all kinds of things can be put inside of these programs to make them run much more easily, actually, for the end user and really get the full use of the instrumentation. However, this requires a dedicated person, someone to take the time to learn this and become expert in using the software. You want to make sure your assay can be scaled down to a 96 or 384 well plate format, or even 1536 now. Not all assays can do that. However, real-time qPCR lends itself quite nicely to this format, and so it's an excellent time to use automation. If we look on the next slide, a lot of times I get the questions, well, what robot should I buy? What you have to consider is not only what your needs are right now today, but also think about the future. What future things might I want to bring and use this robot for? So make sure the features give you all the flexibility that you require. There are robots that have a single tip, that have only 4 tips, that have 8 tips. Some of them have the 96 and 384 well tip heads, like I've shown with the FX robot, or they have tools that have a single tip or 8 tips and work more like an 8-channel or a single-channel manual pipetter. The more tips you have, the faster the job can be done. The span 8 capability, which I'll expand on a little bit later, gives you maximum flexibility and speed. However, using a 96 or 384 tip heads are very fast. However, they're not as flexible as using the span 8, for example. Look at the next slide. When you're pipetting with a liquid handling robot, you have to keep a few things in mind. By default, robot software is set to maximize the speed, but this really minimizes the accuracy. And so you have to learn how to compromise speed for accuracy on a robot to make sure that you're actually pipetting where you want to pipette and how you want to pipette. So when you set the robot for 2 microliters, for example, you want to make sure you're as close to actually dispensing 2 microliters as you possibly can. So the first thing you have to do is slow down the aspiration and dispense steps, and this is dependent on how much volume you're actually bringing into the tip. You want to put in delays. These are important, especially if you use barrier tips, and you should, especially for qPCR, because the liquid needs time to actually come all the way up into the tip and settle before you pull out of the source and move on to the dispense step. One way to get excellent accuracy is to over-aspirate. So I've got 2x here, for example. If you aspirate twice the volume that you need, you dispense half of that volume back into the source, then dispense the single volume that you want to dispense, and then go back and dispense any remaining you have back in the source, and then you keep repeating this. This will ensure maximum accuracy. Sometimes you have to pre-wet the tips. It depends on what the solution is. You have to use an appropriate size tip for the volume. You'd never use a P1000 for a 2 microliter volume, and it's the same as with robots and the different tip choices you have there. It's really best to use food color dyes for an initial program check. you want to make sure that everything is being dispensed into the proper place, especially if you're using a 384-well plate and you have a complex pattern. This can become quite challenging to figure out later on. So by using multiple colors, if you use yellow, for example, and blue and red, you'll end up with green and orange, and you'll know that you've mixed things together properly. Using tartrazine is an excellent way to check for accuracy by absorbance. This is what we normally do, and this is how we find out whether or not our robots are doing exactly what we want them to do. On the next slide, working with 96-well plates. So this is how we started in 1996. We had a 7700 and a Biomech 2000. So we had single and 8-channel tip tools, and we set up our real-time qPCR doing that. By using the single-channel tool to set up an RT reaction, we had plus and minus RT master mixes, 5-log standard curve, and up to 20 samples on a plate. We had to do that with a single-channel tool because the layout was complicated, and I'll show you that in just a second. It took us 45 minutes to set up that 196-well plate and over an hour to run the RT reaction on the thermocycler. We're being very conservative in those days. We added then PCR master mix right on top of that RT. There was a 10 microliter RT reaction, and we put 40 microliters of PCR master mix right on top of that with the 8-channel tool going right across the plate. It took almost 2 hours in for the real-time reaction on the old 7700, and that was state-of-the-art. It took a long time, but it worked quite nicely, actually. Next slide, please. So this is a picture of the old 2000 sitting in a hood, actually, a very large tissue culture hood. In the back, you can see the plate straight back from where the tip is right there. And then to the back right, you can see the tips of the master mixes and the 5-log standard curve, and then the aluminum block in the forefront, and just in front of the 7700 plate, you can see where all the samples are, and then, of course, the tips are over there on the left. So as I said, it took a long time. This robot is still in the lab, and it still works just fine. It's just not as fast as we would like for some of us. On the next slide, you can see the original 96-well plate layout. As you can see, we had a 5-log standard curve. We had triplicates for everything. We had one no-template control. We had the samples in 4 wells. 3 of them had reverse transcriptase, and those are the blue wells. And the green wells, we had a minus RT, or a no amplification control, for every single sample on every plate. We used assay-specific primers. That's what I put the AS primer there on the left in the gray box. We had 4 microliters of samples, 6 microliters of RT master mix. We ran the RT reaction on a thermocycler, and then we added 40 microliters of PCR master mix to give a final volume of 50 microliters on this plate. Okay, next slide, please. Second generation 96-well plate. As you can see on this plate, we have reduced our standards down to duplicates, except for the standard number 5. We still have one single no-template control, and we were able to increase the total number of samples on this plate in the same pattern to 21. Next slide, please. So here's data from the 96-well plate. You can see by looking at the CT values that the triplicates are very good. These robot worked excellent for working this. We did this for 3 years. Next slide. This is a standard curve from the data from the 96-well plate. This happens to be the human calgranulin BLE assay. As you can see, from the red dots that the samples are stretched across the standard curve pretty well. We probably had some nice changes in this assay. And on the next slide, you can see this is from another assay, again set up using the Biomech 2000. You can see that the standard looks pretty nice. We did this over a 3-year period of time for a Wyeth project, and we got some excellent data out of this project. Next slide, please. So working with 96 and 384-well plates, in 1999 we shifted to a TCAN Genesis 100 robot, and that had span 8 capability. The span 8 means that the tips can expand and contract in the y-axis, and I'll show you an example in a picture in just a second. And they can move independently up and down in the z-axis. So this increases your flexibility and what you can do with this robot by a lot. It speeds things up tremendously. Span 8 allows the use of up to 8 tips at once versus only 1 previously. We used fixed tips here. We don't have any disposable tip costs. If you use 5% Clorox bleach and some water, you can clean the tips between samples and you don't get any carryover even for real-time qPCR. This cut the RT setup time to 17 minutes per plate, and that's for a 384-well plate, not a 96-well plate. And believe me, it was worth every cent. The next slide shows a picture of the TCAN Genesis actually doing an aspiration from sample tubes. As you can see, the back 4 tips that are up in the air have already aspirated sample from the 4 tubes in the back that are right next to the ones that are actually in the tubes in the foreground. So tips 8 through 5 are aspirating a sample right now, and you can see that they're sort of spread apart. That's the span in the y-axis. And we go to the next slide. We can see them now dispensing into the 384 well plate, and you see they've come back together again, and now they're dispensing in a narrow format. So they were up and down independently and spanned, and now they've come together, and they're all coming down together. If we look at the next slide, we can see what the 384 well plate layout looks like now for our new setup. We have 2 no template controls at the top left there. We have 5 standards in red. You can see now we're going from the lowest to the highest. We can get 93 samples on this plate in the same sort of layout that we had on the 96 well plate. Again, we're using assay-specific primers. In this case, however, we only have 2 microliters of sample and 3 microliters of RT master mix per well. We do the RT reaction, and just as before, we add 15 microliters of PCR master mix, still using the Biomech 8-channel pipetter, and bringing up the total volume of the 20 microliter PCR on this plate. As you can also see at the top, we can either do a single standard curve with 15 samples, 30 samples, or 45 samples, and then if we get even larger sample sets, we can break it down into 61, 71, and then finally 93 samples. So in the blocks, we have 6 sectors across going left to right on the plate in blocks of 4 wells, and we have that sort of flexibility with each of those programs being written depending on what the investigator has for a sample size. If we look at the next slide, we can see that we can also run 2 different assays on exactly the same plate. The assays look pretty much the same, up to 45 samples on the left side, and then it's reiterated on the right side. We can run 2 different assays. We can also run 2 different sample sets if we have to, and 2 different assays. So this gives us maximum flexibility for the use of the 384 well plate and the robotics programs that we're using for setting this up for our investigators. If we look at the next slide, we can see data from a 384 well plate. This is not exactly what the data looks like coming out of a 7900. We have a macro that processes the data. You can see about 4th or 5th column over on the right, left side, you can see the CT values, and I think you can see that they're all very tight. If you look towards the right, you can see something called CT delta, and you can see the word pass under there. That means that the macro has looked at the triplicates for all of these samples, and they have all passed our QC. And the column next to that says NAC quantity minus CT, which means that the minus RT minus the mean of the plus RT values gives a CT value, and that value has to be 6 cycles or greater or it won't pass QC. In this example of the first 10, they've all passed. But if you notice on the right, sample 31 is in green. That means that sample did not pass this QC test because the DNA concentration was too close to the cDNA. If we look at the next slide, we can see what the standard curve looks like from this exact assay, and as you can see, it looks pretty nice. And so, once again, the robot is doing a very nice job of setting up the assays for real-time qPCR. On the next slide, I thought I'd bring in some information about whole cell lysates, since Dr. Wang talked about it in his talk. And we've done real-time lysates versus purified RNA experiments using the Roche real-time ready whole lysis buffer. What we did was we treated cells with staurosporin or DMSO, which is the carrier for this experiment, for 3 hours. And then we made cDNA from either purified RNA or from the cell lysate. Then we added that cDNA from each culture, 4 cultures for each group, to qPCR cyber green master mix. And then we put that on a tox array from Lonza, Bar-Harbor Biotech, per culture. So we had 4 cultures for each group, and the total number of plates is 16. So this is the kind of experiment where you're loading this up, and you have to fill up a whole 384 well plate with 10 microliters per well of cyber green master mix plus the cDNA. Inside each well on the plate there is a primer pair that is unique to a different assay. So we had, almost 384 different assays on this in each plate. So we loaded this using our Biomech 2000. Again, using the 8-channel tool, we did a quad dispense, so it's fairly rapid actually. If we look at the next slide, you can get an idea of what the data looks like. This is from an LC480 real-time qPCR instrument. On the top, you can see what the purified RNA pattern looks like from a 384 well plate, and on the bottom, what it looked like from the cell lysate. So I've marked where the 18S rRNA is, which is well shifted to the left compared to all the other transcripts. But you can see that the data for the lysate is shifted to the right several cycles, but in fact it looks almost identical to the purified RNA. And in fact, in this experiment, we were able to show that there were 19 significant transcript changes that were shared between the cell lysates and the purified RNA, and they all validated with individual qPCR assays. So automation is essential for setting up this kind of an experiment. So in summary then, next slide, I would like to say that choosing the robot program is important. It fits your workflow. I'm just keeping the future in mind so that you don't buy something that won't work for something that you might want to do in the future. You have to become an expert at using the software, and having a dedicated user is really a must. Robots are excellent for repetitive, large-scale tasks. However, they're not good if the workflow changes often. So and by the workflow, I mean the actual setup of the plates, not necessarily the volumes. Those are really easy to change. Using the same robot for plasmid and nucleic acid preps and then setting up for qPCR on the same robot is not a good idea. That robot is going to become contaminated over time, and you will never be able to get that out of that robot. Always perform QC on new assays before running real samples. Well, that's pretty much. Back to you, John.
Greg, that was a great overview of approaches to efficiently automate PCR workflows. Lots and lots of practical advice for our webinar attendees. Thank you so much. Before we proceed, let me remind everyone of our live Q&A segment that comes right after the panelists made their presentations. Please type your questions into the Ask a Question box on the lower left of your console and then hit Submit. Our third panelist, Dr. David Noor, is now ready to begin his presentation. David?
Thanks for the introduction, John, and thanks also for the opportunity to conduct my first webinar on scaling PCR automation. Today I'm going to be talking about some of the rationale behind why someone might want to automate, also talking about giving some tips on planning and considerations for implementing an automation project in your lab. Then I'm going to talk about just a review of the PCR workflow with respect to automation, and then give some examples of different scales of automation from a single liquid handler, a workstation with just a few instruments, to a fully integrated system. Next slide, please. And just by way of further introduction, I'd like to mention that Agilent Technologies does offer an awful lot of components to help you with your PCR workflow solutions. We offer individual plate sealers, centrifuges, barcode labelers, stacklers, and more. We also offer two liquid handlers, the Bravo and the vertical pipetting station. We offer Benchtop workstations and fully automated Biocell systems. All of these instruments run with Bioware software, and also I'd like to mention that Agilent does offer reagents as well from 5 different PCR polymerases, as well as PCR and qPCR amplification kits and purification and cleanup kits for RNA, DNA, and PCR products. Next slide, please. So if you're considering automating PCR in your lab, one of the main things that most people think of when they think about automating this is the throughput gains that can be achieved, or just maybe you want to be able to handle more plates in a day. This can help you shrink research and development timelines. But another really important thing to consider for automation is the gains in quality that you can achieve with this. Just by implementing a process standardization and uniformity, you can improve the consistency of your reactions. Obviously, this will help you reduce error and also reduce the subjective data analysis. In some cases, it's very difficult to have technicians hand pipette complicated reaction setups, and especially this is true for the case of 1536 well plates. So automation can actually enable this type of a format strategy. Obviously, if you're successful with this, then this can help you reduce your automation costs. And another benefit is just that you'll be able to redistribute the brain power in your lab because, as we all know that have done this, that scientists are very expensive liquid handlers, and this frees them up to do other, perhaps more important jobs. The next slide, please. So when you're planning to automate, it's really important that you understand your process. And there are really sort of triangle of three aspects to consider, and that is scope, timeline, and cost. And that's why I've said in the undertext on the title is to let the process drive. It's really important to hold one of these items and let the others will fall into place. If the most important thing to you is that you keep your costs down, then you're going to have to consider that perhaps the scope or the timeline is going to be sacrificed somewhat to do that. But it's really important in any case for you, when you're planning to automate, to really review your methods, really understand your process, and understand what an automation, implementing an automation will do to your upstream and downstream compatibility and bottlenecks. It might also be a good idea to consider how your system might be flexible. Maybe you can use this for other functions in your lab. But at the end of the day, this is really important because if you have a bad process to start off with and you automate it, then you're going to end up with a bad automated process, and that's not going to make you very happy. So it's really good to put some effort into that. Next slide, please. And when you're implementing this, then it's really good, obviously, to collaborate with a good vendor. This is someone who has good components. They're easy to use, load, teach, monitor, and clean. Their components are reliable. They're accessible by the operator and by the service people, and they're safe. A good vendor will obviously have a good track record, and because they normally have to work with other third-party people to bring about many instruments together into one system, it's really good that they have good people. This includes project management, the engineering talent to be able to do your project. Obviously, software, technical support, and service are all considerations of choosing a vendor. But at any rate, it's really important that you be clear with your requirements with your vendor and communicate with them often. Be a collaborator in this process. Don't just simply send in an order and wait for your automation system to arrive. Identify a super user in your lab. This is somebody who can take that system and really understand it and understand how to really make it stand up and talk in your lab. And then it's also important to use that system immediately. Don't let it just sit around for a couple of months. Start to exercise it, even if it's only to run just a few sort of qualification runs. Next slide, please. Now I'm going to move to talk about to review PCR workflow. There are basically traditional and real-time PCR workflows that are very similar. In a traditional PCR workflow, it starts with purification or isolation of the sample, and then these are formed into reactions. They're thermal cycled. It's an optional step for some people depending on why you're doing the PCR, but a cleanup step can be performed, and then the detection or the analysis is done at the end. Normally in automation, thermal cycling tends to be the bottleneck because the plates that you set up spend more time in the thermal cycler than most of the other steps. In the real-time PCR workflow, the steps are the same up to the setup, but then the thermal cycling and the analysis or detection are done in the same instrument. Next slide, please. So just talking about the first step, which is nucleic acid purification and isolation, or some people refer to this as NAPI. The source material can be from anything biological, cells, tissues, bones, anything like that, and each of those has its particular challenges. But the idea is that once this sample is converted into some sort of a liquid or semi-liquid form, then the nucleic acids can be isolated, usually by some form of affinity chemistry. And the two most popular methods that are used in automation are magnetic beads and silica-based columns. Mag beads usually have something like oligo-DT or some other nucleic acid binding surface on them. And their use in automation requires that you have not only a magnet station, but some sort of a plate handling robot to move the plate on and off of that station. The silica-based columns isolate total nucleic acids and either use a DNase or RNase treatment to selectively give you back either DNA or RNA. They usually require the use of some sort of a centrifuge or in some cases a vacuum, but in either case, if you do this in an automated fashion, then a robot is required to move the plates to those devices, or to, in some cases, to assemble or disassemble the vacuum device. And I mentioned charge switch technology because it's actually available for both magnetic beads and silica-based columns from Life Technologies. There are dedicated automation platforms for nucleic acid purification and isolation, but they rarely perform all the steps, and in some cases the formats may not fit the remainder of your workflow in an automated PCR setup. Next slide, please. When thinking about reaction setups, it's important to determine your process strategy. Are you going from one sample and doing many reactions in a single plate, or do you have many samples that you're performing one or just a few reactions in a plate? You need to consider how you have your plates set up, and I think Greg Shipley gave a very good example of that in his previous talk. So there are other layout considerations for your liquid handler and your system. What are you going to do with tips, reagents, cooling? Do you need cooling on your deck? How are you going to handle your primers and master mix? All of these are considerations that you need to work through in order to be successful. It may be that for your liquid handler that there may be other applications that you're performing that use similar workflows like conventional sequencing, RNAi library transfection, or other magnetic bead purifications that you're using. Not everybody has a dedicated liquid handler that is only used for just PCR setups, so it's good to consider all these things and arrive at the most convenient setup for you. Also, as your plates are being formed, you're going to need to figure out how to cap and seal these prior to introduction into the thermal cycler, so you need to also consider the downstream processing. Next slide, please. So here's an example of a PCR setup where it uses different modes of the head, and it's very similar to the one that Greg Shipley pointed out. In the plate at the top left there on the bottom, there are primers in row 1, a master mix in row 2, and a set of different types of templates in row 3. So in order to begin to prepare the plate, one of the things that you might want to do is to prepare an intermediate plate. And I've used, and as an example, I've used the same plate to do this, but these sources can be on different plates or different reagent troughs. But the example that I'm using uses the same plate. So in the number 1 process, using a single tip mode, transfer the templates from row 3 to the first column, and you make duplicates of these, and then use the row mode of the head to transfer primers and master mix and combine those into row 8. And then in the second one, you aliquot using a row of tips. You aliquot the master mix and primers into a fresh plate, and then using the column mode, you can aliquot the templates from a single column and then transfer those across the plate and eliminate the last column, which is used as a no template control, and so that's how you can achieve the type of setup that's shown there at the bottom left. Next slide, please. And then just to review the plug for Agilent here, that we do offer a number of workflow components that address just about everything in the PCR workflow, from kits and components for purification and isolation, setup, cleanup, and analysis. We do offer a bioanalyzer and a real-time thermal cycler, but currently these are not particularly automation friendly, but we do have those components. Next slide, please. I'd now like to kind of shift to an example of a liquid handler that can be used for PCR workflow. The Bravo is a small, versatile, lab-friendly liquid handler. It can be placed into a hood. It's easy to clean, and it runs VWorks software. It has an optional gripper robot to move plates around on the deck, and we offer a number of different pipetting heads. These are easily changed out by the operator, and they come in 96 and 384 disposable and fixed tip formats. There's also a pin tool that's available through VNP Scientific for this instrument. The Bravo has high accuracy and precision, both with respect to the liquid handling and the head positioning. We can pipette from 300 nanoliters up to 250 microliters. This allows us to get into any SBS plate format up to 1536 well formats. We can, as I've illustrated previously, we can work in columns, rows, or even single tip mode, and this allows us to move from tubes to plates and anything in between. The software obviously handles tip tracking in order to do that. There are also many accessories that are available for the Bravo that are both on the deck and other accessories that you can put alongside them. Next slide, please. But if you're going to move to a more fully automated system than just a liquid handler, then you need to utilize some type of a plate handling robot. And we split these into two different styles. There's a workstation and a system. So shown at the lower left is a Benchtop, which is a plate handling robot that hands off plates on either to the right or to the left at a couple of different positions. And it has integrated with it plate stackers. And so this is used to construct what we would call a workstation, consisting of just a few instruments. And the Benchtop comes in both versions with 2, 4, or 6 plate stackers. But if you need more versatility and more instruments than just a few, then the instrument on the right is our direct drive robot, and it is capable of handing plates off in either portrait or landscape format in 360 degrees anywhere within its work envelope. Next slide, please. So an example of a Benchtop workstation, one of the most common here would be a Benchtop 4R with 4 stackers handing off to a Bravo. In this format, we can handle tips. Fresh tips and expended tips would be in 2 stackers, and then fresh plates and prepared plates would be in the other 2 stackers. It's usually used for low complexity assays or protocols, but there are more complex arrangements possible, and one of those is shown. Next slide, please. This is a workstation for PCR setup that uses a Bravo and the Benchtop 4R, similar to what was shown in the previous slide, but we've also added another Benchtop 6R that hands off to the same Bravo. In addition, there's on the right-hand side of that instrument is a plate sealing device and a plate centrifuge, and also a thermo multi-drop combi for bulk filling there. So that just gives you an idea of the type of complexity that is possible for some of these workstation setups. Next slide, please. But if you really need maximum throughput and hands-off automation, then we offer Biocell systems. These use our direct drive robot, and the center figure shows the robot, and sort of in pink its work envelope. This system is driven by our VWorks software, and the scheduler is an event-driven scheduler. Combines the error handling, and all the third-party drivers are integrated into one system that allows maximum throughput. The system comes with customized protocols, and the limitations are really only space in your lab, your budget, and your imagination. And the 2 standard types of offerings that we have are shown in the extreme right there. There's the Biocell 1200 and the Biocell 1800. Those are just 2 different size formats. The next slide, please. And one example of this would be the high-throughput genotyping Biocell. This uses 2 of our Biocell enclosures with 3 liquid handlers that have magnetic stations and tip washers. We have a plate sealer, a Nexus X peel to peel seals off of plates, and a V-spin plate centrifuge. There's also 4-degree plate storage under the deck, enough for 189 plates, as well as multiple plate stackers. And because thermocycling is often the bottleneck in our PCR process, we have 10 thermocyclers that are available that are robot-friendly. This system is also available with environmental control, both heating, cooling, and HEPA filtration if that's required. And so we can run with this, we can run PCR sample preparation, RT-PCR cleanup, and sequencing-ready amplicons can be produced out of this. And the last slide, please. So this is an example of a system that we made that is capable of producing 1,000 384-well PCR setups in a day from raw samples. It contains 5 V11 robots, 1 Stably robot, and 2 plate translators. There are 6 VPrep liquid handlers and 1 TCAN Evo liquid handler on this, 5 Thermo multi-drop combis, and 2 DRAC equators. There's also 4-degree tube storage and a Biomicrolab XL9 tube reformatting station on here. 5 V-spins, 2 plate locks, 2 V-codes, and this is all run off of 1 computer. So this system is currently in use and runs every day. And with that, I'd like to turn it back to John and answer any questions. Thank you.
David, thank you for telling our audience about the range of technologies available for automating their PCR experiments. I thought your discussion of configuring specific systems will be of particular interest to our webinar attendees. Indeed, I'm looking at the screen. It looks like all the presentations have resonated with our audience as questions have been coming in. So let's get the Q&A show going. The first question will be for Larry. Larry, has PrimeSafe been tested with other polymerases in addition to TAC?
Hi. That's a good question. We have used it with a variety of TACs from several manufacturers. So I shouldn't just call them TACs. They're a variety of thermophilic polymerases. And I should clarify that PrimeSafe is actually a family of reagents where we can tune it to the particular annealing temperature of your reaction, particular conditions of your reaction to get maximum benefit. So I guess that's the brief answer to the question.
All right. Greg, do you find the Biomech FX workstation reliable when moving plates on and off the deck? Can you walk away from the workstation?
That's an interesting question. Yeah, we walk away from it all the time. Having said that, I mean, it is a machine, and machines can have their moments. But when you're setting up a very large workflow, as we do, that is timed and works for over many hours, sometimes overnight, you have to sort of pre-run various and sundry steps to make sure the robot is ready to go and is aligned properly. But once it is, it works incredibly well.
Another question, Greg. Do you quantify those samples that amped beyond the range of your standards?
Yeah, well, here's the deal. When you do assay design, as I am known for doing, I QC my assays over a much larger range than my standard curve fits into, and therefore I know that samples that fall outside my standards, I know when I can use them and when I cannot. And I think this is a critical point in assay design, and this is something that people need to do more of than they do currently. So yes, I do, and I know what the limits of the upper and lower limits of my assays are.
Greg, what do you do if one or more of your standard replicates fails to amplify?
Linda, hi. We take them out, actually. That's why we run replicates. I mean, that's what replicates are really all about. That's why I run triplicates for my unknowns as well, so that I can take one of them out if need be. It isn't often, but it does happen, and the same happens with the standards. Every now and then the robot will have a moment, or for whatever reason, amplification isn't exactly correct. But taking one of your standards out, there's nothing wrong with that, as long as you have one that's fitting the curve. So, yes, I do.
Next question will be for Greg and Dave. So, Greg, you can ask first, and then Dave, you can come in, and then I'll ask you some more questions, Dave. But, Greg, regarding qPCR setup and total RNA extraction on the same robot, don't you think a UV lamp could remove the possible contaminations?
Well, you have to remember, I'm a microbiologist by training, and you have to remember that the UV lamp falls off with the square of the distance. So unless the UV is incredibly close to where you want to decontaminate, it actually doesn't do anything. So UV lamps that are in tissue culture hoods where you think you're decontaminating the surface is just ludicrous, and I've done Serratia marcescens genetic experiments to prove that beyond a shadow of a doubt. Furthermore, you have to remember that UV is incredibly hard on plasticware. So anything that's plastic that gets anywhere near that UV intensity for any length of time is going to start breaking down. So I'm not a fan of UV lamps. David, what do you think?
I would echo that, and the other thing that I would point out is that on most automation systems there are so many nooks and crannies in these things that you have a shadow, you can imagine the shadows being cast. It's not going to decontaminate in there. I think the best way is to get a system that is easy to clean and maintain clean practices all the time as much as possible.
And David, which robotic software allows the most flexibility?
Well, I have to say, this is kind of a loaded question because I really believe that vWorks will allow you to do just about everything. The one thing that is, I would say, is the main difference between the vWorks software and many other packages is that vWorks is while it's able to control individual instruments, it's also very good at controlling multiple instruments simultaneously. In other words, to be used as a scheduler. And in so doing, a lot of other softwares that offer schedulers, they're stepwise, where you have to go, what's going to happen in the first moment, the second moment, the third moment, whereas in vWorks you just say, what's going to happen to this plate, and what's going to happen to the second plate, the third plate, what's going to happen to your source plate, your daughter plate, and it'll just schedule all of that. So I think that's actually a lot more flexible than many other packages.
And Dave, how does the Bravo access inner rows or columns of tips without picking up outer rows or columns?
Okay, so if you can imagine, you're specifying that your one row of tips is to be picked up. It will go to a single row on a tip box and pick that up, and then it will put that away onto an empty tip box, and then that means that the next row that wasn't picked up on that first tip box is now accessible, and so that's sort of how the tips are managed. I hope that answers the question.
Thank you. Well, unfortunately, we've run out of time, but please note that this webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends. Thanks again to the panel for the outstanding presentations. I also want to say thank you to our audience for your attention and for your thoughtful questions about various topics brought up during the webinar. And thank you to our sponsor, Agilent Technologies, who made this event possible. Shortly, we'll be sending you a survey, and we would appreciate your feedback on this webinar. Please look out for it and kindly give us your thoughts, as this will help us to continue to bring you topical and timely webinars in the future. Thanks and goodbye.